Hello, everyone, and welcome to Cascades Analytics Live here on LinkedIn. I'm Alexandre Stepanovic, your host and founder of Cascades Analytics, media and consulting bureau focused on the neurobiology of psychedelics and psychoplastogens. And today are truly exciting times for molecular psychiatry. So we want to stay close. We want to go where the action is, and that's why we want to hear insights from the scientists and the founders driving the next wave of psychedelic drug development. And today I am very happy to welcome Chris Witowski of Psilera. Psilera is developing next generation psychoplastogens for frontotemporal dementia and anxiety disorders. So thank you, Chris, for joining us today. How are you doing?
Good. Thanks for having me, Alexandra, and hello to everyone.
Yes, thank you for joining us. And so why don't we dive right in with a question about how did Psilera come together? And what problem did you see that others weren't tackling?
Yeah, first and foremost, we started the company in 2019. I would say we were early adopters in this up and coming psychedelic neuroplastogen area. And the company was founded by myself and Dr. Jackie Von Salm. So together we have roughly 30 years of drug discovery and drug development experience. We had met during actually our PhD studies. We're both natural products chemists. So it just so happens we found ourselves in the re-emergence of natural products with things like psychedelics, as you mentioned. And we were really focused on two problems. And this was drug delivery technologies. We think drugs like, say, DMT, for instance, that lack oral bioavailability, they're given through injections, really not the most adequate drug delivery technologies for a potent psychedelic. So one of our early goals as a company was to develop new formulations around DMT and similar substances. We have published some research now on transdermal DMT with very high bioavailability, also similar with intranasal. So different ways of getting the drug into the body that we think will have better patient compliance. But at the same time, we were also focused on, as you mentioned, psychoplastogens. We tend to use the word neuroplastogen, in effect, the same thing, these next generation non-hallucinogenic derivatives. And there was some really good data out there from a few of our, we'll say, probe compounds, things like Ariadne, Glyceride, 2-bromo-LSD, these derivatives that act like psychedelics in that they were neuroplastogens. They have very rapid mechanisms to and also enduring therapeutic benefits, which is very unique to this drug class. But they didn't have hallucinogenic effects. So really, in a sense, we created the company in 2019, focused on these two areas. Happy to talk about it more, but that's where we started.
So who is Psilera ultimately building for? Which patient groups are you prioritizing first, and why those populations?
Yeah, it's an important question. As you mentioned, we're focused on frontotemporal dementia, FTD. So it's a rare disease. It affects about 60,000 patients within the United States. Effectively, it's an early onset dementia. These patients primarily onset is psychiatric. So you're seeing mood disorders, personality changes, apathy, largely a psychiatric onset. And then later in the disease is where you see more of the traditional cognitive decline with other Alzheimer's and related dementias. So this is where we're building first with our lead drug, SIL006. But once we prove out this mechanism, proof of concept, we are going to be looking at larger dementia populations. We think the neurodegenerative category is underserved first and foremost, but we think this drug class, these neuroplastogens, and stimulating neurons and helping create new connections, really can have benefits for a lot of neurodegenerative diseases. And also, we think that traditional psychedelics are probably contraindicated in this population. You have high risk of psychosis if you give them a psychedelic drug. I think there's some ethical problems there and side effects that might be more prevalent than in other patient populations. But as well with the cognitive issues in this population, giving a patient a psychedelic drug and effectively having a set and setting profile like we do with other psychedelics, I think is going to be a lot more challenging. So that's why we think this non-hallucinogenic drug is really going to be sort of the best strategy in this category.
Mm-hmm. But there's not one FTD, right? There's multiple. And what is the model you use to target a certain type of FTD?
Right. So within FTD, there are subcategories. So 60,000 patients in the FTD category. About 50% of these patients are the behavioral variant or BV FTD. And these are patients that are severely psychiatric. This is really where we're going to be looking in terms of patient stratification. There is a really good recruitment strategy to find these patients and enroll them into clinical studies. And ultimately, we think we're going to be very strong responders to SIL-006. So that's certainly where we're starting. But once we get some of these signals, we can look to expand beyond 006. We have other prodrugs within our pipeline that we can effectively keep our orphan status in FTD, but then launch, I would say, similar programs in other areas.
Mm-hmm. So you've emphasized dual 5-HT2A and 5-HT6 activation. What's the mechanistic logic for combining these targets, and what preclinical signals convinced you it's worth pursuing?
So SIL-006 is the first in class. It's the first selective partial agonist of both 5-HT2A and 5-HT6, so two common serotonin receptors. So of course, you probably talk a lot about 5-HT2A, so I'm not going to go deep there. But 5-HT6 is probably, not familiar to everyone. And if you dial back the clock maybe a decade or so ago, 5-HT6 was really a promising target for Alzheimer's disease. There were multiple phase 3 programs with 5-HT6 ligands, and the phase 2 results were great. Didn't translate, unfortunately, into phase 3. But we think the selectivity and really the synergies between 5-HT2A and 5-HT6 really bring together kind of a really crucial therapeutic effect. And in our models, what we see is largely the psychiatric benefits from 006 are 2A driven. I think we know that from a lot of the other drugs, clinical studies going on within this class. But also with 5-HT6, the location of these receptors are largely within the hippocampus, which is responsible for memory. And we see very strong, very rapid, and robust procognitive effects. And we believe this is tied to the 5-HT6 activation. So we think, 5-HT2A proving out the psychiatric benefits, that's something we can do pretty quickly clinically. We've mapped out clinical studies with 12-week endpoints. But when you're looking for a disease-modifying drug in dementias, largely this is done through cognitive assessments. And we think that's really where the 5-HT6 benefits will come in. So again, that's some of the reasons why we think this mechanism is really intriguing as a first-in-class drug.
Mm-hmm. What do you think 5-HT6 activation does? It prevents the degeneration, or it helps restore? Or do you have any ideas here?
There's been most of this preclinical work, but neuroplasticity is stimulated through a 5-HT6 mechanism.
Mm-hmm.
The interesting thing too about 5-HT6 is it's only located within the CNS, so you're not going to see other peripheral side effects. So again, I think the procognitive effects, any way you stimulate this target, whether it's antagonism or agonism, you see these strong and robust procognitive effects. So It's still a little to be determined, effectively how and what these downstream signals are. And I think that's kind of not unique to our program, but broadly across these psychedelics and neuroplastogens. But
Mhm.
what we do know is, these benefits are very real based on clinical studies.
Mhm. So back to 5-HT2A, there's an ongoing debate on hallucinations and signaling bias, the GQ versus beta-arrestin pathway. Which paradigms do you think are most convincing, and where do you think the field is getting it wrong?
Yeah, that's a very, very good question.
It could be a
So
whole conference maybe on that, but
it very well could be. I think for us, we're looking at a GQ mechanism.
Mhm,
Primarily, this is where the first gen or classic psychedelics are activated. With beta arrestin, the theory is that this does not lead to hallucinations, although I have seen some research that points otherwise.
mhm,
But what you do see, especially with repeated beta arrestin activation, is receptor desensitization, which also leads to basically drug tolerance, which we don't think is beneficial, especially for our drug in this population. Um, yeah, I mean, in terms of benefits, I would say most of the clinical evidence is through a GQ pathway.
mhm,
So that's kind of where our focus is.
mhm, mhm. GQ pathway, but not hallucinogenic, so it requires a different, it invites a different theory, probably receptor occupancy or presynaptic versus postsynaptic, or maybe some other mechanism at work here. Um, so why don't we dive deeper into frontotemporal dementia? Uh, so you mentioned it, but could you explain what's the hypothesis linking psychoplastogens, especially the 5-HT6 activity, to disease mechanisms or symptom relief in FTD?
So let's just take the word frontotemporal. So this disease is primarily focused in the prefrontal cortex, unlike other dementias, which are largely hippocampal. So when you look at the prefrontal cortex, it's exactly where high receptor densities of 5-HT2A are.
Mhm,
And this is also why you see a strong uh, psychiatric issues within these patients. And there is a known pathology with 5-HT2A in these patient populations. So I think first and foremost, it's a receptor pathology. It's a brain circuitry and location. I think there's very strong evidence there with our drug.
mhm,
And when you're looking into the symptoms of these neurodegenerative disorders, there are two very good approved drug case studies to follow. First and foremost is brexpiprazole. It's a 5-HT2A antagonist for agitation and dementia. And you also have pimavanserin. It's a 5-HT2A inverse agonist
mhm,
for the treatment of psychosis and Parkinson's disease. So
mhm,
there's a very strong link between activation of 5-HT2A and broadly the symptoms within this neurodegenerative disorder.
mhm,
And when you're looking at dementias, about 80% of any dementia patient is going to struggle with a psychiatric disorder, whether that's depression, apathy, anxiety. So 6 million dementia patients, 80% of them have a psychiatric disorder. Again, it's a big unmet need. And we think there's really strong clinical evidence there that we can prove out rapidly with our clinical development plan. But we do have hopes longer term that with the activation of 5-HT6, with the benefits on cognition, that we can prove this out as more of a disease modifying drug. And that's something that we plan to do in later studies.
mhm. And 5-HT6 will be acute as well? Or
In terms of dosing frequency, or
yes, in terms of dosing frequency, yeah.
so that is something we're actively trying to parse out
Mhm,
in our preclinical models. Ultimately, we think human patients are the best way to understand the dosing frequency.
mhm,
But looking at things like EEG, sleep patterns is another area that we're investigating pretty heavily.
mhm,
So I would say our drug is going to be more in an intermittent slash chronic dosing regimen.
mm,
We've seen anywhere from every third day to once weekly dosing is effective with our drug in preclinical models.
mhm, mhm. So So that's my question. What endpoints or digital biomarkers would give you early confidence in FTD?
I would say maybe backtrack just a little bit,
Yeah.
but one of the areas we really like about the neurodegenerative category is patient stratification. There are genetic markers that we can identify patients. There are known pathologies within the brain. So with an FTD, you see brain shrinkage in the prefrontal cortex, very hallmark signal of FTD. So we're very confident that we can find patients that have the disease, which in psychiatry is a little bit more difficult. I would say, we're not quite there yet, but the work in neurodegenerative disorders, even with the approval of liquid biomarkers, especially in Alzheimer's disease, we can use those as exclusionary, which does help us too. But in terms of looking at therapeutic biomarkers, EEG, I mean, it's one of those that a lot of people are investigating. There's strong evidence to show that the drug induces similar signals to a classic psychedelic like psilocybin, but you're not seeing sort of the alpha wave changes that you do with a psychedelic drug. So we think this is a really non-invasive way to show the effect of a drug and changes within the brain. I mentioned some of these biomarkers. So neurofilament light chain has come on leaps and bounds, especially for diseases like ALS. Actually, FTD shares a lot of similar pathologies with NFL in them. So we're looking at those liquid biomarkers. As I mentioned, sleep, a lot of our preclinical work has shown that we can reduce a lot of the sleep detrimental qualities in tau models. So this is pretty ubiquitous across most neurodegenerative diseases.
Mm-hmm.
And we think by helping sleep, we can really help a lot of the other cognitive processes, psychiatric processes. And of course, those are things that you can track very easily in clinical studies with wearables and others. And the last area that we're looking at is even facial expression recognition. There's a strong link in this patient population to look at eye tracking and show some of these psychiatric benefits that aren't purely a questionnaire model. So these are some of the areas that we're looking at, others that I can't quite go into too deep of detail. But again, we really like this aspect of neurodegeneration because of these biomarkers that currently just don't exist in psychiatry.
Will you be able to prescribe, will CL006 be prescribed to any moment in the education or early, late FTD?
So we're working with a really strong clinical advisor, Dr. Elizabeth Finger. She's literally the world expert in the psychiatric symptoms of frontotemporal dementia. And a lot of the research has really pointed that, as I said earlier, the psychiatric symptoms are really early in the disease of FTD. So we are looking at patients earlier on in their disease. And I think ultimately what a lot of drug developers in this space are coming to realize is that early treatment, early diagnosis is really going to be crucial, like you see in oncology and other areas. So I think getting to these patients early is really going to be where we can see the strongest benefit from our drug and ultimately help them live a better quality of life.
Mm-hmm. So if we focus on CL006, could you tell us in one sentence, more or less, the chemotype, the mode of action, and what differentiates it from first-gen tryptamines?
So as I mentioned, CL006 is a first-in-class partial agonist of serotonin 2A and serotonin 6. This is the first drug that we have seen across any of these drug class that has this specific mechanism. Mm-hmm. As you had pointed out, receptor occupancy is kind of where a lot of people look at, is this drug hallucinogenic or not? And there's a lot more research coming out. We're working with Dr. John McCorvey. He's really an expert at sort of this serotonome, I think as he's calling it. Mm-hmm. And, basically, you can overactivate your drug into 2A, and that's where you get more of the agonistic psychedelic effects. Mm-hmm. If you tip the scales the other way, you get an antagonist, which are, still effective drugs. They're antipsychotics. There's many, many instances of these being, therapeutic drugs, commercial drugs. So really with our drug, we're kind of in that sweet spot where we can induce a rapid neuroplasticity mechanism, but we're not inducing the psychedelic side effects. And the last thing that I'll mention, and we spent a lot of time on this, is reducing some of the off-target effects. And this is related to 5-HT2B with cardiovascular concerns and also HERG. So we've really created a what we think to be a safer drug within this class that ultimately can be non-hallucinogenic and dosed in more traditional paradigms, not under clinician supervision for hours and hours.
Great, especially if it's a chronic treatment. Any PK/PD features, onset, half-life, brain penetration, you can share that map to specific clinical use cases?
So our drug is oral, very strong oral bioavailability, over 65%, which we think is great. Again, looking at that take-home model, I think an oral drug is an ideal target product profile there. In terms of half-life, it's similar to psilocybin, but what we know is the therapeutic effects, they're not correlated to the pharmacokinetic effects. So certainly we see a longer durability of effects beyond the half-life of the drug or the plasma effects of the drug, which we think is important. And lastly, strong blood-brain barrier permeation gets into the brain very rapidly and to a high degree, which for any CNS drug is really crucial.
Mm-hmm. I'm wondering if neurodegeneration might prevent, bioaccessibility somehow. But this is maybe a different topic. Is there anything you'd like to say on the ongoing development of SIL025, your compound for anxiety disorders?
Yeah, I would say broadly, when we started the company, we were focused on this tryptamine drug class. And there's specific modifications that we can make to these drugs, which number one, were novel, which are important for developing composition of matter IP, which is very crucial for a biotech company like Psilera. Mm-hmm. But also we've created a huge chemical library, and we've been able to screen this through in silico technology. We've really included some unique methods there, including dynamic simulation. So we're not looking at a static drug and static receptor, which you're not going to get the best results there. We've also incorporated water into the active site of these molecules. So we feel very strongly that we can translate what we see through computer and technology into at least animals at this stage in producing non-hallucinogenic drugs and being able to predict this early on so that we're not blindsided later on in our preclinical development pathway. So SIL025 is one of these drugs that had come out of our screening. It is a new chemical entity, and there's a lot we can do to this molecule in order to improve drug-like characteristics. It is, I would say, number 2 in our pipeline behind 006. We have seen very strong and rapid anxiolytic benefits in preclinical models. The drug works through a 5-HT1A mechanism, which is commonly known for anxiety disorders as well as depression disorders. And I would say, our preclinical screenings really first and foremost focus on pharmacology, because we believe this has the best translation from preclinical models into human models and therapeutic effects in clinical trials. So yeah, that goes a little bit under the hood of what we're doing, but we have a lot that we're excited for beyond 006, 025. So yeah, thanks for asking.
Sure. So now if we move to translation and development, what does your translational funnel look like, from in vitro signaling to in vivo behavior to early human biomarkers? You touched some of these aspects already.
Yeah, so like I said, pharmacology is really where we started because we know there's a strong translation. So we were looking for drugs that activate 5-HT2A. This is the common ground for neuroplastogens. So we think activation of 2A is important, but again, not tipping the scale of receptor occupancy into a psychedelic drug. So that's where a lot of our screening goes, and this is performed initially in our platform, our computational platform, which we call Psilera Third Eye. And we screened hundreds of thousands of molecules, including a few known neuroplastogens at that time, known psychedelics. So we knew exactly what receptor interactions, what orientations of the drug would produce a 5-HT2A agonist, but not a hallucinogenic 5-HT2A agonist. So this is where we spent a lot of time kind of honing that model, increasing our predictability, which I will say right now, there's not been a single drug that we've put into animals that has been hallucinogenic, and this is what we've predicted all along. So we feel like we have a really good feedback loop there.
Mm-hmm. And you use EEG now to see that the alpha waves are disappearing, and
that certainly that comes a little bit later on in the workflow. I mean, initially, we test our drug in silico, then we go into animals. Okay, are we inducing a head twitch effect, which correlates to a hallucinogenic drug? Okay, that feedback loop is there. What other phenotypes are we seeing? So we can run some psychiatric models just to get an idea of where the drug potentially has therapeutic value.
Mm-hmm.
And then we can go into more detailed work, in vivo work, like we've done with 006 and humanized tau models, and looking at specific cognitive deficits, sleep deficits, and also doing some in vitro work, looking at iPSC models to show specific circuitry of the drug, long-term potentiation, which links to neuroplasticity, and as well looking at inflammatory markers, which we think are prevalent across a lot of CNS disorders. And what we know about our drug class is they're very potent anti-inflammatories. They get into the brain. So we think by targeting a specific pathology and things that we can look at as a liquid biomarker in humans, really increase the likelihood of that translatability into the clinic.
Great. How are you approaching regulatory path, scheduling risk, CMC, abuse liability assessments for non-hallucinatory or low-intensity compounds? Because one of the advantages of psychoplastogens, it's also the lack of hallucinations for the FDA approval, for example.
Sure. So we're working with very strong CMC partners, and they liaise directly with the DEA. So at least with 006, they've confirmed that this is a non-scheduled drug, which of course is a lot easier from a regulatory perspective to manufacture and store this drug. So that's great. And again, goes to your point of these next gen molecules having benefits over the first gen psychedelics.
Mm-hmm.
In terms of clinical development, I would say a strong portion of our phase 1 in healthy volunteers is going to be looking at dose-related effects. Are we seeing psychoactivity? We're going to be using 5D-ASC and other metrics that are used in psychedelic drug trials. So for us, we're looking at, of course, the lack of these psychedelic effects. But that's really, I would say, an early proof of concept into the translatability from what we've developed in our preclinical workflow into the clinical workflow. Certainly from there, we're going to follow the FDA's guidance, abuse liability and things like that. But initially, a lot of our work is going to be focused on proving that these drugs are non-hallucinogenic. But I would say, we have strong conviction right now in that the head twitch model, the pre-pulse inhibition models that we've ran are quite translatable. So we don't expect to see any hallucinatory effects or dissociative effects in humans.
So now in a crowd, would you say that the landscape, the psychoplastogen landscape is getting crowded right now? And what's Psilera's sharpest edge, target profile, IP, delivery, clinical strategy, all of the above?
The neuroplastogen landscape, I wouldn't say it's crowded. I mean, when you're looking at things like GLP-1, I would certainly say that space is crowded.
Right.
But, number one, there's a huge unmet need within psychiatry, within neurodegeneration. So there's not going to be a winner takes all in this scenario. I feel very, very confident in that. One thing I'll mention a little bit as an aside. So actually Spravato reported their sales numbers today, roughly on a 2 billion annual run rate with treatment resistant depression. and they're only penetrating about 3% of the market. So, again, there's not going to be a winner takes all. I think from a competition standpoint, there's certainly a lot that still needs to be done. I don't think it's crowded. I'll just stop there.
Yeah, but the Spravato is the price point is very high, isn't it?
Yeah, yeah. I mean, it comes to reimbursement. I mean, frankly, I've interacted with people who've had trouble getting into Spravato treatments because, again, the reimbursement is challenging. I
Mhm.
think pricing the reimbursement of a drug is something that's quite simple. I think that part is covered, but
Mhm.
then getting the in-clinic aspect, the nurse, the doctor time, I think that's where the challenges are. And again, goes to show, if you can develop a take-home treatment, a lot of those things have already been solved in terms of a commercial profile, which
Yeah.
I think is where, pharma's interest really is. But again, a big unmet need for any of these treatments. And I think, the next couple of years will certainly be a very interesting space to watch. But going back to Psilera and our differentiators, I would say, at least to our knowledge, we are the first company to really plant our flag in the neurodegenerative category. Most of these other psychedelic neuroplastogen developers, they're mainly looking at depression, maybe anxiety, but largely the psychiatric category. So we think, number one, that's a big differentiator for us. As I mentioned, we were an early adopter. So we founded the company in 2019, started filing our patents. So we have a very strong IP portfolio. Uh, I would say millions, if not billions, of new chemical entities are under patent currently, and issued through 2041. And this number is going to keep increasing as we continue to solidify our IP strategy. And one of the earlier things that I had mentioned about drug delivery technologies, we had actually closed a deal earlier this year with Atai Life Sciences, and they acquired our DMT patent portfolio, so around drug delivery technologies with dimethyltryptamine. So again, I think we have a very strong IP strategy. We're leveraging that in a lot of ways so that we can really focus and double down where we feel very strongly that neuroplastogens are going to be sort of this next generation wave that, it's like any drug development pathway. You have one drug that makes it to market. I mean, we'll say Spravato, right? This drug gets to market, and now you've got a slew of NMDA companies focusing on, less dissociative derivatives. You're going to see the same thing with psilocybin when it's approved as a 5-HT2A agonist. Okay, what can we do then for the next generation of drugs to make it more palatable, a take-home drug, fewer side effects, fewer cardiovascular risks? So, it's going to take us a little bit longer to prove it out, but we think we're in a very strong position with our IP and given our clinical development differentiation to really, I think, play the long game here.
What milestone should we watch for in the next 12 to 18 months?
So certainly with 006, we're focused on getting our IND submitted by the end of next year. That's our current focus, is proving out that still 006 is safe. We've done some initial toxicology models, and we see a strong safety profile with this drug. Actually, one of the things that we just announced a couple weeks ago was NIH funding towards alcohol use disorder. We do have some initial uh, preclinical data to support a very strong, um, abolishment of alcohol drinking with a SIL006 derivative. So
Mhm.
we think there's another area that we can sort of play off of. And, we're kind of running dual paths on both FTD and AUD, that we think SIL006 effectively could be a pipeline in a product like drug. So I mentioned FTD, other dementias, AUD. We think really 006 for FTD is the tip of the spear.
Mhm.
So really the next 12, 18 months is going to be focusing on getting this data and getting the IND submitted, and we should be running clinicals the early part of 2027.
And where do you see Psilera? My last question: where do you see Psilera? Sorry, in 5, 10 years, things go to plan.
Yeah, so I'll do one better. So our strategic initiative for the next 3 years is to have 3 clinical stage assets by the end of 2028. So I mentioned our DMT patent portfolio. So this is covered with an ET product. So that's our first clinical asset. 006 will be in the clinic, I would say, in a little over 12 months. And then, we're also looking at our pipeline, like you mentioned, Silo 25 and other drugs that we have there. Really where we want to be is a clinical stage company, first and foremost. I think the strength of our pipeline is really the strength of our team and our company. And as we continue to build on this, that's where the rubber meets the road, is showing this clinical benefit. Focusing purely on the science is where we've been since foundation. The ET deal that we closed earlier this year, I would say certainly won't probably be the last licensing deal that we do. Certainly we're looking at potential licensing opportunities, co-development opportunities within our pipeline. So, this gives us the flexibility to then take those funds and reinvest them back into our pipeline and platform where we think we have really strong capabilities to generate new clinical development candidates in a de-risk fashion. So I think, and I'll close with this, the next 5 to 10 years is going to be transformational in terms of brain research. It's not crazy to think that in the next 3 years, we could have maybe 3-4 psychedelics, not including ketamine, on clinical or commercial drugs. And again, this helps the entire field. It helps patients more than anything. And, I think we're definitely in the right place at the right time, doing the right things. So who knows what the future has in store, but yeah, I'm really excited for it.
Yes, me too. And well, Chris, thank you so much for your time and your insights. And hearing from you about the work ongoing at Psilera is super interesting. So thank you very much.
All right, thank you, Alexander.
I'll see you next time. Thank you, everybody, for tuning in. And I hope you enjoyed this conversation with Chris Witowski of Psilera on SEAL 006 and SEAL 025. If you have any question, please send me a message, or if you would like to have the transcript of this conversation, you can contact me as well. Until next time, be well, and we'll see you soon.